Okay.
Okay.
Very good morning to all of you. Honorable Minister Dr. Jaikar, Vinayaka Mission Research Foundation deemed to be university. Dr. Prashant Bode, Managing Research. Dr. Honorable Rajshekaran, Dr. Vishnu Bhatt, supervisors, faculty members, delegates who are joining this particular one-day workshop on good laboratory practices and clinical laboratory practices. I'm extremely happy that amongst his busy schedule, our Honorable Registrar has agreed to inaugurate this workshop because he himself is a practicing pharmacologist. So he knows the importance of what GSP is all about. And I hope this one-day workshop will be very, very useful. Just one word about good laboratory practices. We are living in a scientific world where we take the research of what we are doing in the laboratory and we conclude certain things based on that, be it science, be it medicine, or anything for that matter. So, good practices are very, very essential in every part of in every facet of life, and certainly what when we are working in laboratory practices. and I'm happy that Dr. Prashant Bode has agreed to conduct this workshop. Now I request Honorable Registrar to inaugurate this workshop.
Good morning to all. It gives me an immense pleasure to be with you all today for participating in this workshop on good laboratory practice and as well as good clinical laboratory practice. So the workshop is hosted by Vinayaga Mission Research Foundation, the deemed university, which has been established in the year 2001, and actually the Vinayaga Mission's Research Foundation, that trust has been constituted in the year 1981, and first of its institution, Pharmacy College, started in the year 1982 with 7 students, and now it has grown as a deemed university with 13 constituent units and 7 schools. Around about 12,000 students are now they are studying in various faculties like medicine, dental, engineering and technology, pharmacy, homeopathy, and arts and science. All the faculties, around 12,000 students and about 10,500 faculties are working in this organization. So in this organization, since we have mainly on health related institutions, this good laboratory practice and as well as good clinical laboratory practice is very much essential, not only for the faculty members and as well as the public also to know about this good laboratory practice and as well as good clinical laboratory practices. Why this laboratory practices is to know much about is the nowadays based upon the good clinical laboratory practice, the diagnosis is done by the our physicians as well as surgeons. So this good for a laboratory or for a good laboratory, not only the machines is not only enough. The machines should be, it has been calibrated properly and it has been maintained properly. Not only the calibration and maintenance and working of the equipment, but there is a very good knowledge, thorough knowledge about the equipments, and the results prediction also very important for a good clinical and as well as good laboratory practice. So it has been felt it is much essential for each and every one of the health professional to know about this good laboratory practice and good clinical laboratory practice, so that they can update that knowledge on these 2 areas, so that they can practice well in these areas. So for that, for with us our Prasad Bode, the director Clinic Minds, has been accepted our invitation to be with us to engage ourselves from morning till evening on good clinical laboratory practices and as well as the principles of good laboratory practices. Sir, on behalf of Vinayaga Missions University, we welcome you for participating with us and highlighting on principles of good laboratory practices. In this juncture, I thank Dr. Annabel Rajashekaran that who has been invited me and who has given me an opportunity to inaugurate this workshop on behalf of Vinayaga Mission University. At this juncture, I convey the wishes of our Honorable Chancellor Dr. A.S. Ganesan as well as the Vice Chancellor Professor Dr. P. Sudhir for success of these workshops. So once again, I am very happy, glad to inaugurate the workshop on good laboratory practices and as well as good clinical laboratory practices. Thank you very much. Thank you for giving me an opportunity for inaugurating this workshop. Thank you, madam.
Thank you, sir. Thank you very much, sir. Thank you so much for being with us, sir. Thank you.
Thank you, madam.
Thank you, sir.
Now I request Dr. Annabel to introduce formally the speaker, Dr. Prashant Bode.
It is indeed my proud privilege to be part of this nationwide workshop, which is being organized by our prestigious university. And we are so happy to have the Honorable Registrar with us this morning. It's my proud privilege to introduce the speaker of the day. And the speaker is Dr. Prashant Vidyadhar Bhode, and who started his career as a pharmacist, and he started working as a research fellow in the Indian Institute of Toxicology, Pune, in the year 1988. Dr. Prashant Bhode rose to the position of Director, Clinical Development Center, Advanced Center for Treatment, Research and Education in Cancer, ACTREC, Tata Memorial Center. He then went on to become the Associate Director, Clinical Operations and Office Head in Sri Lanka for Quintiles. Later, he became the Director of Operations, Medanta Duke Research Institute, MDRI, in Gurgaon, Haryana. And currently, he is the proprietor, principal consultant, managing director of Clini Research. Clini Research is in Bombay, and we are very happy to note that Dr. Prashant Bode's PhD was in pharmacology, in clinical pharmacology at the prestigious Sheth GS Medical College, KEM, which he did in the year 1998. So, Dr. Prashant will be taking two sessions, one on good laboratory practices, which is mainly aimed at the laboratories for research, and it will also, and then he will go on to present good clinical laboratory practices which are attached to the clinical departments, that is the paraclinical basic medical departments, pathology, microbiology, biochemistry, and so on. And this will be in the afternoon. There will be a 10 minutes break around 11:30 when he finishes a part of the session. He will continuously present on good laboratory practices starting from now. I'll be handing over to Dr. Prashant. At this point, I'm grateful that with the blessings of our founder chairman, Dr. Shanmuga Sundaram, and our chancellor and pro-chancellor, vice chancellor, and the registrar of the university, we are able to conduct this nationwide workshop, which is attended to by participants from more than 100 institutions, from medical colleges, dental colleges, pharmacy colleges, and nursing colleges. Now over to you, Dr. Prashant.
Annabelle?
Yes, sir, Dr. Prashant should start, sir, right now.
No.
This guy has to put Dr. Prashant.
Dr. Prashant is online? I think he was there. He was there. He got disconnected, I think. Just to give a ring to him.
No, I'm not able to see anybody at all, sir. So in this.
Give a ring to him.
Yes, I will.
He might have got disconnected.
No, he is there. I'll call him.
அன்னைக்கு 15 மேல ஃபுல் மே வந்தது.
We request all participants to kindly mute, because in this there is a large gathering, so we are not able to mute you centrally strictly. So we request all participants to please
டைரக்டா ஹேண்ட் ஓவர் பண்ணியாச்சு.
Prashant got disconnected, so just please, we'll wait for him to join again. Have you joined, sir? Hello? Hello? The panel is பிரிக்காததால
You are able to speak to him, Dr. Annabelle?
I spoke to him, sir. I can't even see where my icon is. I am unable to see. It's
No, that's okay. No, he has not joined it. He has not joined it.
Hello.
Dr.
Yes.
Prashant, can you hear me, Dr. Prashant? I read
Hello.
to him, sir. Hello, Dr. Prashant?
Yes.
Can you hear me, Dr. Prashant? வேற யாரோ கௌரின்னு வேற பேசுறாங்க.
Dr. Prashant Bode?
So, but what happened, sir? You're not on. I can't see you at all. If it's not even showing how many people are there.
No, that is showing 420 people are
Yeah,
there.
420. But you know, quite a few are not able to join. They're saying so. It's not allowing him to join when he says join meeting. So I have sent.
Jai Shri.
I've sent you his phone number too, sir.
Madam, good morning. Ask few people to go out of the group so that he can join, then ask them to join. That's the easiest way.
No, he has joined. He has joined. He has joined. He has joined. Prashant Bode has joined. Just hope for. Just wait for a second. Yeah.
Ma'am, sir has joined, ma'am.
Yeah.
Yes, he's there. Dr. Prashant Bode is here. All right. Okay.
Yes, he has joined.
He has joined. Dr. Bode, can you start the presentation? I'm sorry, we didn't realize you were not. And the inauguration is over. I have done your introduction of the facilitator as well. So it is time for you to speak, sir. Can you hear me, sir? Dr. Prashant?
Ma'am, Dr. Prashant sir has to connect to the audio. It will
So
take.
the audio you have to connect, sir. You have to connect the audio, I think, Dr. Prashant. It's not connecting, I think.
Who is the host? Mahalakshmi?
Dr. Satyamurthy is holding that Mahalakshmi position, sir.
Okay, sir. That ask him to for the Prashant Bode to give the screen sharing and all the facility.
He
So
tried
that
everything, sir. Before the meeting, he was seen, everything was there. Suddenly it got cut. And now he's not able to join, I think. I have given Dr. Satyamurthy, Dr. Prashant's phone number. Maybe he can guide him to join.
Satyamurthy
No,
sir,
I don't have
actually,
his phone number.
I just now sent it. I sent it, sir.
Satyamurthy sir,
In
who
the
is
WhatsApp.
Yes, sir. Yes. Yes, sir.
actually who is doing in AVIT? You call him and ask him to, huh? Lakshmi Mogan.
Like, yes, sir.
I ask you keep in touch with him and find out how to
Yes, sir. Yes, sir. Yes, sir.
include Prashant birthday.
No, it is included, sir. This is for everybody. Yeah. Join now. No, not yet. I'm giving a ring.
But
Prashant sir, please. Now you can. You are connected.
Can you hear me, Dr. Prashant?
Yeah, I can see you. Okay, now,
Yeah.
I don't know, some technical issue. Can you hear me now?
We can hear you, sir. You have to share your
Okay.
screen. Then we can start.
Screen, yes, I'll share my screen.
என்ன மேடம் வெளியே தொல்லையா பெரிய
Can you see everyone?
Not yet.
It is loading, sir.
Not yet, sir. We can see right now, sir. Right.
Okay. So, good everyone. And, sorry for this technical glitch. I think we have to face such unknown somehow or the other. But okay, we have to overcome all this as well. Like we are done
Yes, sir.
with. Okay. Just 1 minute. So today's topics of interest are good clinical laboratory practices. Okay, is it gone? Oh God. I'll reshare once again.
Sir, we're not able to hear you.
Can you hear me? Hello?
yes, sir. Yes, sir.
We are able to hear you, sir. Please go ahead.
Okay. Just a minute. I mean, I have to restart my presentation. Something happening here. I don't know what it is. So far, I never experienced something like that. Just 1 minute. So I'm pulling some time before I can start. The program is loading. My name is Dr. under Dr. Kshisagar at GS Medical College in subject of pharmacology and clinical pharmacology. And after that, I worked in industry for a long time, almost 25 to 30 years. And I started my consulting firm about 6 years back. Work in the field of toxicology, clinical pharmacology, clinical trials, bioequivalence trials, pharmacovigilance, and pharmacology. So we will have our talks in
Sir, we are not able to hear anything.
Sharing the screen because the file is too big. It is taking a time. Just hold on.
அவரும் நிறுத்திட்டாங்களா? Participants, please stay with us.
option. Okay, I think I'll share the screen. That is easier. So, okay. But one and then now, and can anyone see? I mean, can you see now screen? Okay. Now loaded, no? Yeah. Fine. Okay.
As well as your figure is also visible.
It's fine. You please start.
You need to mute all.
Sir, go.
Yeah. Please
Okay.
go ahead.
Question and answer session. We can take it offline only later on. So questions listen to Dr. Rajashekara within 2 days after the session is over, and I'll address queries by email. So the end and certificates will be after the quiz completion. On is able to hear me. That's what I presume. So what are we actually? We are a CRO. What kind of CRO? We are a contract. We may be a person, or we may be an organization, or we may be a university. Our academic or a combination of these two, and we are contacted or contracted by sponsor. Their trial related or research related functions, and we work in the various field and analytical lab, clinical chemistry, etc. So there are multiple fields we work. What GLP? I mean, GLP refers to good. Refers to a system of management controls, which is meant for laboratories and research outcomes. Consistency and reliability of results. See, this is outlined in the OECD principles of good. Of course, many other national and international regulations and guidelines. So it is a quality system concerned with an organ and the conditions under which basically it started with non-clinical health and environmental safety studies. The definition says that the quality system concerned with organizational process and the conditions under which environmental safety studies are planned, performed, monitored, recorded. So these are basic functions. If those who are familiar with the good clinical practices for good clinical practices, design, conduct, plan, or whatever other things. Basically, originated probably, or which were based on this definition of GLP. Back GCB started, but however the principles are much parallel. So let's see what is a quality system. Quality system is a policy which is planned for execution within an organization for a purpose, and the purpose is of course and repeatable and accurate results. Then such system is planned in detail. Functions are planned and implemented in operations over a period of time. This system is then later on performance using quality control and monitoring. And once this is set, the system is set, it undergoes an audit whether original policies which were meant with an objective, which were prepared an objective are being met or not. Of course, this audit is conducted by various people for various purposes, so the objectives and but then internal audit here I'm specifically referring to will undergo the lateral, will undergo management review, and why the policy was started in the beginning and why it was implemented and how it implementation is appropriate or not. And there will be, of course, gap analysis. Then again, a program of continuous improvement will be implemented, may get revised, amended, and this is a circular thing. It goes on and on. So there is a perspective about GLP is that it is an instrument and tool to ensure. It is not. But it is based on scientific aspects, and it's an instrument and tool to ensure the quality, which means honesty about the data, that is information, and it is about business basically. Why we time and money and efforts and all the things, all the work we do, it's business. Otherwise, why would we go? I mean, why a sponsor will come to a CRO if the sponsor is not sure that can guarantee? So this is a GLP is a pre-purchase warranty. We can say that okay, the work will be done while the work is being done and work is completed. So it's a practically a business perspective. But of course, it is not. This is about science. So this is a science of business and it's a business of science. And it is concerned about well-being of human being. Necessity is for funding, for getting sponsored projects, for our own general perspective of to well-being of human being. And this is strictly not a checklist line item. Especially, I would say this is important from the management. Okay, they don't have to say that we have done the GLP compliance, but this is a mentality because it requires from the top in the beginning, and it is a continuous support. It is not that okay, we have completed that checklist, and this applies to even hierarchy. So this is not a checklist item. You tick mark, you've done it. Now that's it. But it's a continuous process, basically. So what GLP is not? You see, GLP is not directly concerned with the scientific design or content of study, science in itself, but it is scientific at the same time. The scientific design may be based on test guidelines. For example, we have a kinetic study which is based on guidelines which are published by various regulatory authorities. Judge any scientific value like the drug regulatory authority. GLP does not judge that, but adherence to GLP will. See, in most of the studies, we have designed a protocol to control the variables, and these variables are a source of error, or which will actually stop the from or maybe a hurdle. It will be a hurdle for us to conclude those variables are controlled, and with the GLP as a tool, this control becomes very well easily by adding data which is collected during study or test or whatever. So our focus today is GLP and GCLP. Understanding of right now, first part is GLP principles, then improving knowledge of GLP principles and its regulatory basis. Mention about components of GLP compliance. This is not in detail about GLP compliance, but it is about the GLP principles in details. So discussion on study plans and data quality and integrity auditing, etc. This is, and there will be overview of GCLP on the similar lines in the second part. So there will be introduction. I think we can skip. Of GLP is non-clinical safety testing of test items contained in the pharmaceutical product, or veterinary drugs, or feed additives, or industrial chemicals, or maybe it is a testing test items to and or the safety with respect to human health and or the environment, or expanded scope since inception. Applications of GXP is being done, which we will see later on. See, all toxicology labs and data is generated falls under GLP. And in fact, that is true for everyone. Where regulatory submission data is generated, the GLP mandatory. All analytical and bioanalytical labs, they support pharmacokinetic and bioavailable. All clinical chemistry labs where they support the patient care in for the patients participating in clinical trial, the cell balance. And as I was saying, like in currently there is a good in vitro lab practices labs also. If we are going to in a research lab, we are going to generate data which will be submitted eventually to in a maximum possible limited possible manner. We should implement GLP because many times GLP right now at the research level at prior to submission. So any research which is not submitted but there as well, the credibility of data goes away, goes high. And so like last but not least, any are done. So background, okay, in why how this GLP thing started, in our that some of the regulators, including US FDA, they investigated a number of cases of poor practices in toxicology laboratories throughout the same. And this investigation was done in about 40 plus laboratories, and there are aspects which were not thinkable at that time, even at that time where controls were not so stringent. And poorly managed studies, insufficient training of personnel, in case of there were deliberate frauds. And in those days, the safety and efficacy requirements for a pharmaceutical product were very, very different. Earlier, even the efficacy was not required to, but later on this, of course, amendments came, and lots of regulations started developing even around 40s. What did FDA find? So they were poorly trained study directors. So a person. There are some, of course, there are some comments which I am going to make in with a great sense of humor. So to, exact meaning. Say, so person who is very close to the say top management, we may become a study director. He may not have a requisite qualification, but it becomes a study director. And there will be study personnel which are not appropriate, having appropriate experience or even capabilities. It's poorly designed protocols because the objectives of protocol, but just doing a tick mark study so that we have done the studies. Protocols may not be followed because they may be expensive to comply with. So procedures were not conducted as prescribed for whatever reason, either by at the QA level, or maybe at the project manager level, or maybe even at the study director level. So whatever, and the raw data is correctly identified without traceability. So later on, when inspectors come for verification, so we thought we don't require it later on. So, but we have conducted the study. Those were the answers which were received earlier. So data was by responsible persons. There is a lack of standardized procedures. There are SOPs did not exist for many aspects or at all completely. In animal husbandry, there is a mix-up of animals. There is no segregation. There is no age control. There is no demographic things which were happening. So there was inadequate characterization of test items and systems. So we prepared. So there was no uniformity in these test items as well. So how the was the question. So there were inadequate resources. The people were not there to conduct the study at all. I mean, so there eventually it meant equipment is not properly calibrated or otherwise qualified. See, this is more easy having a viscosity meter, which is not. See, there are multiple methods of measuring viscosity, and each applications. So using one cheapest available tool for measuring something different, which is measure which is not meant for it, is not going to serve the purpose. So then reports are not sufficiently verified. They do not or there is inaccurate account of studies. There are many places where the data which was never conducted included in the final reports. Inadequate archives and retrieval processes. So these were all the reasons for genesis of GLP. Experiments were poorly converted. I think we saw this, but anyway, like I'll just okay. See here again the management perspective, which is a critical review of data, or proper supervision of the personnel. So this is a very important aspect from the management's top management perspective. Not be given for the scientific qualification and adequate training of personnel involved in the research studies. So sponsors also fail to monitor studies performed the whole or in part by the CR. So there is no adequate control by sponsors. So the firms also fail to verify the accuracy and completeness of the scientific data in reports of non-systematic manner before submission to FDA. So there was no review of the data. So it started the New Zealand and Denmark started good laboratory practice in 1972, and FDA earthed the frauds which were generated by toxicology labs. They started a process of implementing GLP. They're like, so what happened in industrial biotest labs is a very famous example. They received a tip of problems with the test submitted to FDA, and there was data which was unbelievably clean. I mean, they're too good, and there was like we say in all cushy, no gum. Management was there. I mean, there was no rats on a 2-year study which developed a cancer. Like there are some default cancers which are all documented and proven to be not connected with the test compound, but they are probably connected with just or maybe resulting because of the trauma which is faced by the animals. But there were multiple visited. They found that safety tests were falsely claimed to be done, but none were performed, especially the final ones were to be proven to be normal. Records were so poor that data could not be pieced together what work had been done. There was no proper collation of data. The terminal blood and urine samples were not collected. So there was falsely done and results were written in the final report. The draft data tables for the blood and urine samples, of course, were blank because there were values with a signature of a technical writer's name and thus approved. So FDA proposed then GLP in 1976, and the final rule came in June 1979. The Environment Protection Agency separately, which is also USFD, US government organization, issued another draft in 1983. So we have our own example, but that is a little bit latest as well. So it has 2010 in Mayapuri radiological, which is labeled as Mayapuri radiological incident. Working in Mayapuri presented with a radiation sickness, and they visited some. I think it was which hospital? I'm not very clear. I think I missed. Visited a hospital, and hospital fortunately sent alert to the Atomic Energy Regulation Board, which oversees usage of like what happened exactly. The radiation from cobalt-60 has affected these workers, but how did it enter? By AERB, the cobalt-60 radiation actually originated from the Department of Chemistry. This gamma cells, which were manufactured by a Canadian company, were being used and certainly retired in 1985. Of course, nobody took care of these aspects later on, and these cells were auctioned by the Delhi University officials and maybe from the Department of Chemistry people. 2010, and the scrap dealers worker dismantled these gamma cells, and it exposed almost 10-15 people. Cobalt-16 pencil sources caused an unwarranted high exposure, and 7 injured and 1 person dead. So later on, of course, the action started. Show cause notice were issued, suspension. All the activities those were using radiation sources in Delhi University were stopped, and there was a lot of only after the compliance with all stipulated regulations was done, the University of Delhi was reissued the license for. They were not allowed to handle high activity sources. So actions under the Atomic Energy Act were taken. Charge sheet were filed against members, and also under section 337 and 338, the causing hurt and grievous hurt by act of endangering personal safety of others, and 304A for death due to rash and negligence act of IPC. So only one of them, and is not actually connected, but we need to ask this question. This is self-inspection or what introspection questionnaire. See, are we honest in our day-to-day work? Very simple question. I mean, we train people, or have I trained myself to do the honest work, or work which I know I'm really capable of doing it? Do I know I'm doing something which is I believe is honest, or I know that I'm not doing honest work, whatever may be the case, aware of consequences of my actions. So am I working, and this is especially true for most academic and this is not geographical this is not geographical just to generate publications or doing worthwhile data which is useful for humanity, or and see, why would people do such thing? Why would scientists would do such thing if the performance criteria and promotions are going to be based on quantity rather than quality? Actually, the scientists are going to do what is required. So this is a question to the management. I mean, quantity or quality? Any more important than somebody's life? Or do we have enough training program to tune people to do authentic work? Okay, so this is one present slide. I'm not going to do this again, but only once. So control for addressing all the issues we have related environment, and we have ICH guidelines, then from Code of Federal Regulations and multiple guidelines they have. And then we have trial directives and guidelines. Then we have environmental protection agencies. We have OECD. Then we have good clinical practices, clinical laboratory practices, manufacturing practices, good analytical laboratory practices, and whatnot. In addition, there are local for target registration and place upon. Say we want to submit data to Japan, or we want to submit data to Australia or USA. We have to comply to requirements as well. In addition, there are national regulations and guidelines in our case, for example, ICMR or Drugs Controller General of India, and sometimes even municipal zone who have their own guidelines and rules for waste management, for example, who have their for employees, and these are to be complied. I mean, besides all scientific. So compliance to CGLP, CGCLP, GCP, GMP. C is meant for current and applicable rules, regulations, laws, and guidelines. So we work, for example, we work in a testing laboratory, clinical laboratory. So we have a GCLP. So if I am working in a which is clinical ward doing clinical studies, I'm an investigator. So for me, GCP is applicable. If I'm working in a manufacturing is applicable. But what does this mean really? I mean, all these guidelines which are represented by circles, smaller, bigger, what case may be. Am I supposed to do this in isolation, or it is a combination approach that I have for GCLP? Now, GLP and GCP are implicated, or these principles rooted in GCLP, but does still bit, or I have a global approach. We have to have a global approach, because to be is being human. Sometimes there may not be a mention about other regulatory guidance, or but they are inherently meant to be compliant. For example, in all these guidelines, there is one practice which we call as a good document. So it is not generally mentioned everywhere what are those principles of good documentation practice. So basically we have those as well. So say for example, there is now one more latest guidelines is good in vitro IVR. So now this is one of the latest which was introduced, I believe in 2017. So the scope of the good practices, good X practices is going to be higher and higher. Why do we have to comply? To promote the quality and validity of test. Whatever test we are doing, whether it is a short term, it's a patient glucose level monitoring testing, a big program like carcinogenicity test, which runs for 2 years. We have to provide quality and validity of the data. We need to be compliant, therefore, for a reliable, repeatable, and auditable, and of course acceptable data. Our acceptable by concerned regulatory authority, and even by the sponsors. And when the sponsors are trying to, the public also have to accept that data. There is a need to build a necessary intrinsic scientific values. We cannot just create a examples. No, many people, many companies are claiming that they are developing vaccines for COVID. They are developing X product for COVID, but I think they are just taking people for ride. Sometimes understand scientific value built in that product, being from the field, but public do not understand till they are actually by the time many things. So it's of course an organizational requirement, because if organization is not following this compliance, it is, there are many examples which happen in last few years, few decades, and not only in India, all over the world, the organization permanently, they have affected multiple products. Say, for example, there was one CRO in Hyderabad, which went down, and it took down 700 pharmaceuticals with it, and the products which were registered by based on the studies done by that CRO. Question, and this, the sponsor had to repeat the studies, and there were a lot of ripples even as well because of the compliance which was being questioned and intent being questioned and many other things. So it is of course organization of course is represented by the management and the top management. So it is a top management responsibility, and indeed it is mandatory we are submitting the regulatory data. So because we are dealing with safety and efficacy and quality of a product or a test, by doing some tests or a small test, a big test, but we are contributing to a product for a safe and quality aspects for the safety and well-being of human being. So let's and fundamentals aspects, quality and validity of data and results. See, it helps scientists to obtain the results that are repeatable, auditable, and recognized by scientists worldwide. So this is an important aspect, that they are not, again, like say if same experiment is repeated somewhere else, it should be able to repeat the with the accepted variable limit. So aims of GLP is a set of organizational requirements, organizational system for implementing GLP. See, this is for not to assess the intrinsic scientific value of a study. The basic aim is to make incidence of false negative very obvious. So when we get false negatives, sure that it is a false negative. To make the incidence of false positive also more obvious. So when we take whatever results, whether negative or positive, we should be automatically become sure about it. This promotes also international recognition of the study data, mutual acceptance of what is called as mutual acceptance of data. And by doing this, we limit the, by controlling the resources required, by controlling the allocation of resources, we actually limit the waste of resources, because on the contrary, we may buy instruments which are for a particular project, which may be very high value instruments. They were not required, but we have purchased. Things, incidences will not happen, and we ensure high quality, comparability, mutual recognition of. So it stipulates the conditions. LP stipulates the conditions for organizing studies and scientific value. Again, repeat, this slide is purposely repeated. But there are 5 basic points of GLP. We need to mute other people. Since there are resources, which means personnel, of course, resources are one of the most important human being, facilities and equipment, and there is a characterization about the test article and the test system, which is this article. Then there are rules which are actually defined by protocols, study plans, rules, guidances, manuals, etc. Then there are the results in the form of raw data, final reports. There are amendments and many other ways. And of course, there is one most important, which is of the GLP, is a quality assurance. It is of course by quality control or monitoring or auditing. These are the aspects of quality assurance. Let's now look at the resources, personnel, facility, and equipment in short. The GCP pillars are again the management very much behind the GLP endeavor because it is not free. It is very, very expensive to dance with GLP. In fact, because India signed into GLP, the cost has come down heavily. Cost earlier it used to cost multiple crores of rupees before India started GLP, but now that has come down a lot. And of course, the cost for implementation of GLP that still remains the same, but which were very expensive now have come down. And of course, the study director, the quality assurance, the national compliance monitoring checks the compliance for GLP and gives issues certificate to the organization. So let's start with this is not directly, but I have included this over here, and of course, thing, but it is not in this perspective which I have added. The test facility management means the persons who have formal overall responsibility for the organization. See, for example, here we can say the chancellor of the university, or vice chancellor of the university, or dean faculty, or head of the department. They become the top management part, and they are directly responsible the test facility according to the principles of good laboratory practices. So, what data and head of the department will get, he will pass a shortened version to the dean of the faculty. Dean of the faculty will again his higher authorities, but in a way, everyone is involved, and everyone is responsible for promoting science in terms of good science and a good organization. Let's see what kind of personnel are involved. I mean, it's a study point of contact for the particular study or a particular test, a particular scientific endeavor for which we are going to organize at the organizational level. The quality assurance who is going to help the top management, and quality assurance is going to assure and ensure the compliance on behalf of top management, and scientists who will be controlling the data while it is being collected or store the data appropriately after the studies are completed, and the study person who actually conduct the studies, and all these people require equipment which will be provided by the top management. Good science means careful definition of experiment. So, for example, there are a lot of noises being made about useful for only patients who have no serious infection of COVID, and then later on claims are being conquered COVID. So, so design the experimental clinical trial was designed and implemented for asymptomatic patients, and claims are being made for symptomatic and severe patients. Science based on so these are many times these scientific principles are found in reference articles or they are issued by the regulatory authority. and this should be done. Like, there is a controversy related to drugs controller involvement, ministries involvement. So whosoever is right, who has the authority, so they have developed, and all these are controversial things, but I'm giving examples over here just to highlight that science based on known scientific principles, control and documentation of experimental and environmental variables. See, all those variables which are going to affect the conduct of experiment, thereby the outcome of the experiment, thereby affecting the credibility of results, should be appropriate by. So careful and complete evaluation of and reporting results has to be done, and it's the management's response. Of course, they are going to do this through QA, and results becoming the part. They should become part of accepted scientific knowledge at an appropriate time. I mean, until that happens, the work carried has no meaning to the general public or humanity. Good organization, that is again a top management's responsibility. So they have to have a proper planning of studies and allocation of resources. They start, say, 10 studies in a laboratory which has capacity of only 3 studies, or people only for conduct 10 studies cannot be started over there. So provide adequate facilities and infrastructure of qualified staff. For example, a particular scope is required and it is not present, then at least provision should be made that it is outsourced in time, and of course the mass spectroscope also is of the right configuration being done. Then definition of staff responsibilities be done. See, like no one should be saying, no, this was not my part of responsibility. Not responsible. I was not knowing that I am supposed to do this. All this situation should never be happening. And of procedures to ensure proper conduct of studies. Again, this is through document system. There has to be a document that is called as quality manual, which is at the top management level, and of course, which should what is the quality policy of the organization. Then what are the rules under which we are supposed to work? These are, of course, maybe work related, only work related. See, for example, if a person is supposed to be there in the lab every day at around 9, minus 10, 15 minutes or half an hour, then he's supposed to be there. So these are also rules which affect, or if a person is required to be particular day, is supposed to be there at 6 o'clock in the morning because there are samples to be collected. So all these procedures and compliance by the staff be ensured by the management. And there is, of course, system of good record generation and keeping and organized in the, and all this implementation of verification processes for study conducted and results have to happen. So management will ensure that principles of GLP, they are the authority and they are the initiative people who have to state that GLP will be compulsory for everyone. So they need to find the person who is responsible for the management and who will GLP compliance program in details, and he'll be the point of contact for top management till this are being implemented. So then the management has to ensure that there is sufficient number of qualified people, appropriate facilities, equipment, material, and people are of appropriate qualification at the same time. The equipment is, things are available for the timely and proper conduct of study. So there has to be a maintenance of record in terms of qualifications, training, the experience, the job description for each professional and technical individual. So in a project or in a study, their job description has to be maintained. I mean, that is a job description. A person actually has to do much more than that normally. Personal understand the functions that they are supposed to do, and it is management's perspective and duty to provide training for this. If a study person says, no, I never knew that I am supposed to do this, is actually not a failure of that personnel. It is a failure of management that it did not the appropriate training, or maybe person was absent on the day of training and he was marked present. So so within the system, as per records, the personnel has completed training, but the person says, no, I never knew that I was. He has never attended the training. So appropriate and technically valid standard operating procedures, we call SOPs, are established and approved all by top management. Again, here the top management's perspective is important, because see, why only lower people cannot sign SOPs? They of course they're supposed to be written by the people who are actually going to conduct the studies, not by the QA. Normally it's a person. No, QA never writes SOPs, but they administer the SOPs in the system. And but the top management has, at least not on all pages, but at least on the top page, that this particular SOP is now live, going live on particular date after the training is completed. Because management is responsible for the operations happening in their institute or their organization. And if a who doesn't want to do a particular thing writes an SOP which is in contradiction with the objectives of the facility, the organization, then it becomes difficult for management to catch him actually, because there is which says, okay, you do this or you don't do this, and they are being followed, but which were contrary to the objectives of the organization. Should never happen. That's why SOPs are to be signed by the top management. I many times I've seen that top management is this job, but of course there is a delegation happening, and we can't expect the vice chancellor of approve SOPs of all the department. I mean, he will not do any other work on in that case. So he has to delegate this work, say dean or maybe the HOD through proper delegation process, and the HOD then signs the SOPs active in his department. Then there will be some institute related SOPs which can be signed by the head of the principal of the faculty. And there is has to be quality assurance program with the designated people. We cannot have other people just come and do this audit today or just do monitoring today. No, that cannot be happen. There has to be a designated people, and they are responsible for performing all the duties like audit and compliance reports and issuing even the certificate in accordance. The replacement of a study director should be done if needed, and this documented very well, because there are many times the person retires or he changes a job, and then there is need director. There are many times multi-site studies, and out of there are of course multiple study directors. Then one of the study directors also educator, or whatever name you can give as a chief study director or whatever but he must be appropriately trained, experienced, and qualified to supervise the studies and all the study directors. And there of course has to be communication channels which are established. Same thing with principal investigators replacement. There could be change in name, and then it has to be and appropriate approvals and intimation to the regulatory authorities concerned should be done. The top ensure that study director has made approved study plan. That is, we call it is in a protocol, and a study plan depending on a type of study. So it is made available to quality assurance personnel. The mental file of all SOPs, like all versions of SOPs. How did it start? Which is the version which is active? And there is one particular tool, Atron, when I'll elaborate on SOPs. Then individual is identified as responsible for management of archives, the maintenance of mastership, the studies. There should not be a clash that one person who is probably maybe working on 2 or 3 different projects has to be present at the same time, different location, or has to perform a task which is time consuming for 2 or more studies. That should never happen. So test facility requirements, they meet the requirements of the appropriate the use in the study. There are multiple studies, and we have the requirement of say 1,000 syringes, and we have available only 50 500
Please,
syringes.
can you hear it? Can you hear?
So, hello. I think there is a lot of disturbances. Somebody's. So, in a multi-site study, again, the clear lines of communication must the principal investigator, and in addition, quality assurance program, and in case of where collaborative work is required as well. If study personnel are not required to be communicating with each other, then that channel may not be activated. So ensure that items are appropriately characterized. This is particularly true for multi-batch test items or multi-batch, and where repetitive batches of test or reference items are going to be tested. Single test and reference item are also to be characterized in an appropriate manner, but then there won't be the variable aspect statistically defined for reference items. So establish procedures system are suitable for the intended purpose and are validated. See, like for example, now we have required, and there is a Vista operating system available in the laboratory will not be acceptable. And only one official one. There can't be 2 or more competent systems. So in a department, there are 2 or in a commercial organization, if there are 2 people who don't get along with each other, prepare their own schedules, is not acceptable for whatever reason. As an approved SOP, this system has to be how this study master schedule will be maintained, and the responsibility of its and updating are defined by management. Generally, I believe this part will be handled by QM in most of the organizations, but it can change. The various schedules are approved and maintained in the archive as data. So once a master schedule gets updated, the original previous version, the master schedule becomes, then the current master schedule becomes the current, and it is in use. But other versions are archived immediately, and they become done. And of course, once there is a change, the distribution has to be done in an adequate manner, and key responsibilities are needed. So allocation of resources and time, what it contains, the management ensures that allocate and other resources to the specific studies and support areas. See, for example, there are multiple multi-institute studies, and there are one to be carried out in one institute and another in another. So there has to be a collaborative work. How this will be scheduled availability of staff personnel, respective staff personnel at both the institutes. All this has to be managed, and all studies, whether contracted or in-house. So we have to exactly where multi-centric studies are involved. Everyone or concerned people are supposed to know what and when is where it is going, a thing is going to happen. So they are aware and they come and make the material or report or whatever is needed for the next team to start their work. Control procedure is in place to reflect the shifts in the dates and workload. So this happens many times and is a very common occurrence. And documentation, writing, review, and report preparation, they also should be included, because protocol writing takes a lot of time, and it is not just a template which needs to be each and every line in a protocol needs to be checked every time. Even if, say for example, bioavailability studies, most of the times have a standard template and very few things change, and that is really a dangerous practice in a way. Although it is it is an ease of, but each and every line item must be checked and reviewed by QA and by other people. So suitably size, construction, and location. See, the GLP doesn't specify that you need to have 100 square feet, or you have to have a distance between two beds of at least 3 feet. GLP doesn't suggest anything of it. You apply your knowledge, you apply the relevant regulatory guidances, you apply local regulatory norms, design your facility for the requirement of the study, and they ensure the validity of study. GLP is concerned with that. The adequate of the different activities to assure the proper conduct of studies. If there are multiple studies, there should be no mix-up. There should be confusion about what belongs to what or which study should never occur. And also here cannot get mixed over here for different studies. Number of rooms or areas for isolation of test system, especially those suspected of being biohazardous. So we cannot conduct two studies, for example, handling radioactive material, ordinary drug, cannot happen in same project, because those people, those personnel who do not need to get exposed to, will get exposed to the biohazardous material. And the facilities for handling test and reference items should clearly define the stored, retrieved, or dispensed, and transferred, and again taken back to the archives. How the handling and disposal of waste would happen, like the material in the link. Apparatus, material, and should be valid. The system also should be validated for use. For example, there is Excel validation system used for the generations and for controlling environment factors which are relevant to the studies. For example, we need to monitor the temperature of refrigerator, storage area. We need to monitor humidity content of and temperature of storage area. All this monitored and corrective actions taken in case of any issue. So they should be appropriately designed and be of adequate capacity. We cannot have samples coming in and lying outside the facility because there's no place to store the refrigerator. And this aspect should be periodically inspected. The equipment be cleaned and maintained and calibrated. Have to be calibrated. Their temperature monitoring system has to be calibrated, and any repairs or should be maintained. The calibration should be appropriate and be traceable to the national and international standards of measurement. I mean, whenever request for even for a thermometer or even temperature monitoring system, how it is calibrated, who is calibrating entity is itself authorized to do. Is it an ISO certified or NABL certified? So apparatus and material used in the study should not interfere adversely with the test systems. Naturally, I mean, this is a very common sense. Reagents and solution should be labeled to indicate identity, and concentration is appropriate. Expiry date and specific source labeled on the container, a primary container, and if required on secondary and tertiary container as well. Information, course, preparation date, and stability should be available. So if there is a preparation or dilution required later on, how that should be done should be available as a part of SOP or work instructions, but it may be extended on the basis of document evaluation or analysis. Can especially happen for the test material. The expiry date may get extended based stability studies, and this has to be appropriately extended to everyone. Now, who is the main pillar of this GLP organization? The overall organization of test facility, and there will be an organization chart prepared by top management of the facility, which will have right from top management to the will be updated on an ongoing basis. And organization chart is normally the first document which is requested by inspector. And during routine internal audits, the organization chart should be the first document. It gives an idea how the facility functions. See, organization chart forms a part of the quality manual as well. And so when there is a change in organization chart. So does it really mean that, okay, I have a turnover of around 300 employees, and every month 3 people leave and go, or every week 3 people leave and 3 new people. So do I need to update organization chart on a daily basis? Of course not. It can be done on a monthly basis based on the requirements. The study director is directly connected to the sponsor. It is management. It is connected with the quality assurance unit, and it is of course a source of information for the contributing scientist. So it's a pivotal role of study director. It is a single point of study responsibility for overall conduct, and of course the data contained in final report, the archival of it. Responsibilities include, but they are not limited to, but there can be much more than this. These are just the surface or scratch list. Approval of study plan and any amendments to the study plan with which is authenticated by a dated. The quality assurance must have a copy of the study plan, and any amendment should be prepared, certified, authorized in a timely manner, and distributed also in a timely manner before it gets implemented. Get with QA direct in an effective manner so that there is no lacunae in especially the critical communication required for the conduct of studies. The study director has to ensure that study plans and amendments of SOP are available to the study personnel. There has to be training and removal of old SOPs before made available to the study. It's a simultaneous process of removal and addition of new SOP. Ensure that the study plan and the final report have identity and of the contributing institute. Then there is of principal investigator. There is a defined role of the study director at each institute level, and any test facilities or test sites involved in the conduct of studies they are going to do must be clearly defined, and study director has to ensure that this happens at an appropriate time in the. So study director also ensures that procedures specified in the study plan are executed. They're followed exactly appropriate meaning. And okay, I have not added those slides which I normally add, but then plan, and there has to be implementation spirit and in has to be there, just not the spirit, just not the letter of the document. So both can actually can still conduct a fraud. I mean, people can still conduct fraud, but that should not be there. So the intent has to be there, and the study director has to assess and document the impact of any deviation from the study plans. See, deviations are going to be part of the, and provided they are authentic and genuine, they are assessed for their impact on any study plan. Quality and integrity of study and the results, and there has to be appropriate corrective and preventive actions taken if necessary. The deviation should be approved, acknowledged, or rejected, or whatever by appropriate SOPs. And of course, they need to be communicated at all levels, required levels, in a timely manner. See, needs to ensure that all raw data generated is fully documented and recorded, and it is archived later on. The computerized system study have been validated. The data which is computerized has a stamp of date and time, login, and there is proper change control available for such things. There is a requirement of sometimes that CFR, which also is ensures the change control in terms of login and secure password and for the changes. Then sign and date final report to indicate that has acknowledged the acceptance of responsibility for the validity of the data, and to indicate with these principles of GLP. Of course, study director can never say that I say, okay, I'm compliant for 75%, 25%, I'm not. That is not going to be there. He has to say that I'm compliant and and at the same time, I mean, both the way things. And of course, this compliance, whatever statements will be checked by appropriate systems of quality or regulatory side, and they will certify that yes, what you say is correct or wrong. So ensure the study director has that the study plan, final report, raw data, and everything, and materials and control samples are archived. So PI has to ensure the phases of study are conducted in accordance with the applicable GLP principles, and all study directors across all the sites have complied and complete their work on time and as per schedule and as per the requirements and etc. Let's come to study personnel's responsibilities. They must be eligible in those principles which are applicable to their involvement. However, this doesn't happen in isolation, because a person who says that I'm handling only archives of documentation, so I will just move that part of GLP. It generally doesn't happen. The person required to be compliant with all of GLP, or should be knowledgeable in the all parts of GLP. Of course, he will be compliant work-related practices, but he has to be knowledgeable in all the principles practically. See, he has to have to access the study plan and appropriate SOPs, at least those SOPs which are applicable to his involvement in the study. Of course, because it's for entire organization and there are multiple parts of it. So all people cannot be knowing all SOPs. SOPs has to as per their job functions. And there are multiple documents. Say, for example, there is a quality manual. There is a quality organizational rules. There are SOPs. There are work instructions. There are rules. Say, for example, you cannot eat your laboratory. So these are rules. I mean, these are actually not scientific rules, but for whatever reason, the top management has decided that there is a canteen available, and you cannot eat food inside the laboratory. So it has to be compliant. You cannot deviate from that. So, and lead to serious implications because of, say, food particles which are leftovers or which are put in a dustbin can breed in the laboratory, and which can be detrimental to the entire system. They can actually cause havoc in the. So any deviation from this instruction of SOP protocol should be documented on PI. They are responsible for recording raw data promptly and accurately and in compliance with these principles of GLP. So they should not document anything which did not happen and they think that has happened. So they are therefore responsible for the quality of the data. See, they need to, for this, ensure that they are in a healthy condition to minimize risk to the project, the risk to the any illnesses. For example, in current situation, appropriate example for this point, and this ensures the integrity of the study as well, because if many people in the facility get infected, the study timetable and its integrity and its collapse actually may collapse if many people get infected because of infection. So study to the appropriate person any relevant known health or medical conditions in order that they should be excluded or replaced for the operation, not if not permanently, at least maybe for partial amount of time. Quality assurance is a very important aspect of is it quickly, and then we can go for the next part. I am okay with this without any break.
We can have a break, I think. But how much longer will you take before lunch hour, sir? Depends on that.
Be there 1:30.
People do need, because they logged in at 10 o'clock, so it's now 2:15, 12:15. So
12:15, yeah. So by another 1 hour we should be finishing. Actually, increase the time duration. Yeah. Okay. So we'll continue. I think that continuity also has an important one aspect. Okay. So this is and GMP systems, and I'm just highlighted this slide to show importance of quality assurance is there everywhere. Wherever there is a GXP, let it be laboratory, manufacturing, clinical, these systems can never function without the presence of quality assurance, and they are actually management. They are not reporting to the study director. If you can see over here, see in GMP there is no particular of qualified people. It's not a one person's prerogative in GMP as it is in GLP, but QA. So the test facility management is responsible for study director, activists, and QA. And study director in turn is responsible, which we just detailed in many aspects. So quality assurance is a pivotal aspect which ensures quality GLP or GMP or whichever lab practices we are going to have. So there is a revised consensus document. Is already inculcated in the original document now, and there is a QA and management link. So management of a test facility has responsibility for ensuring compliance. But do they do everything? They don't do it practically. They are not supposed to do bench top things. But they have to delegate that. It has their own functioning areas. So, but they have to ensure the compliance. So for that, they appoint effectively adequate number of people, qualified people, experienced people throughout the facility, especially those which are quality assurance functions, or which sometimes which they eventually learn. They have been conducting the study pattern, get promoted to the quality assurance function, which is also acceptable norm. The managers ultimately are responsible for GLP. They should be clearly identified. Manager ultimately is responsible for GLP be clearly identified. Again, this is coming. Okay, managers appointment of appropriately qualified people for both the experimental program and for the conduct of independent QA function. So the person has to really understand QA function and the experimental program as well. And the person must have direct access. So direct access is a very important terminology in especially in GCP, which is used quite emphatically, but it has meaning in GLP as well. To the different levels of management, to top level of management for test facility, because he has to report anything which is not healthy or which is happened or something, some incidents happen which can be critical to the validity and reliability immediately to the top management. And for this, of course, the QA has to have access to all the data, all the study personnel, study director, and other QA fellows. See, the program needs to assure they are in compliance with the GLP principles. How the work be carried out by designated by and directly responsible to the management and who are familiar with the test procedures. A person who has never done an in vitro process, for him, he has to learn, at least he has to get trained how the in vitro processes are being done. The person has never done a particular work, but he has to undergo a virtual training in order this procedure in totality, and he should be able to understand the issues and how execution where can go wrong. So it has to be appropriately, because for example, if there is a small surgical part in or a person who doesn't understand say mass spectroscope has to do quality assurance activity on that. These are the common occurrences. So one person cannot know all, so at least there should be basic primary training given to the person. These people should never be involved in the conduct of a study, even if they understand what's going wrong. The responsibilities include, but are not limited to, maintain copies of all approved in the test facility and ensure that they're available to the corresponding personnel. Master schedule. They have access on up-to-date data which is being collected. They have to information required for compliance with GLP. So there should not be any terms or any of the document, including study protocol, which is in contravention to GLP principles. To conduct inspections to determine the inspection should inspections by QA should ensure that study plans and SOPs are available to study personnel and they are current and people trained and the SOPs are being followed. That is insurance by quality assurance. And he has I'm not going to go in too much of details of these aspects because this itself is a 2-day two-day training program. Even for study directors' roles and responsibilities, we can definitely spend a day, entire day just for it. Otherwise, you it thing to be studied in detail. But see, there are study-based inspections, there are facility-based inspections. There are what FDA does is a for-cause inspection. There is just routine inspection. There are uninformed inspections that you will find an FDA inspector standing at the gate at least half an hour before facility opens, and he sees exactly what is happening in and around. Even such surprise inspections are also happening. Whereas there are pre-planned inspections also happening. Records of all inspections need to be retained. QA needs to inspect the final methods, procedures, and observations are accurately and completely described, recorded. And the reported results are to reflect the raw data of the studies. There has to be a complete match. There cannot be plus or minus. Report any inspection results in writing to the management and to the study director and to PI. But of course, the primary response and study director and PI communication is in parallel, not the primary one. Prepare and sign a DOS statement, which is to be included with the final report. I mean, there has to be a report that I have inspected these studies, I have monitored this, done audits on these particular dates, and all these audits, all the issues were resolved. We call it corrective and preventive action, and the specified types of inspection, the dates and phases of studies inspected. Inspection results were reported to management. All these statements are to be part of the quality statement. In this also will serve to confirm that the final report reflects the raw data. Let's come to the test system. I mean, in a shortest possible manner, but this is again, I'll tell you the most important function of a study. We need to mute other people. Let's come to this system. Okay, there could be apparatus which is used for generation of physical or chemical data, for example, mass spectroscopy. I'm again referring to this, my favorite instrument. Suitably located and appropriately designed and of adequate capacity. So you cannot use MS 1000 for metabolic studies because it's not going to detect those metabolites. You need a higher 3000 or 4000. So integrity of physical or chemical tests must be ensured. See here, integrity has a indirect implication. Validation of the system in terms of calibration of the system, in terms of IQ, OQ, PQ. So GLP, but by merely mentioning the integrity of physical and chemical test system, it it all. Sometimes if you say many things in and or specify too many things in a particular guideline, complies organization complies to those requirements, then the regulatory authorities cannot pinpoint very easily. But when such statements are there, integrity, it covers all. Actually, it covers what is said and what is not. So biological test systems, for example, there is a biological assays, or there are animals. They are part of biological test systems. They should be maintained for storage, housing. Since GLP initially was written for statements are more appropriate for those conditions. So they should be established and maintained for storage, housing, handling, and in order to ensure the quality of the data. But if you extend this to any other system, the principle remains the same. Source, data arrival, conditions, etc. should be maintained and monitored. Receipt record should be including characterization, data received and used during studies, how it was used, how it was replaced, everything should be documented. So for pharmacy, in a pharmacokinetic study, we have a pharmacy even in clinical study. Material is received from the sponsor, then it is actually stored at in an appropriate storage conditions after that said quantity has been received, and then how it was dispensed to several patients, and if there is a written how how it is processed later on, and if there are any remaining quantities, how they are actually stored, how they are disposed past or expiries, or we cannot do any testing on such samples, how they are discarded, sponsor, whether they are incinerated, everything is actually pre-planned, and identified in order to homogeneity and stability are ensured to the degree possible, and contamination. So two different studies, test article or reference article should not get mixed up. The storage container information, like expiry date, sponsors, specific storage instructions, etc. Batch number, purity, composition, concentration, or any other characteristics to appropriately verify and identify test items which are subject to the study, and the study specificity should be the stability data for under storage data should be known for all the studies. I mean, it should not happen that we're still using it. Subsequent preparation, see, it may happen in a particular study that stability data, earlier the stability was given only for 3 months, and later on it was 6 months, later on it was 1 year. So there is a procedure for all the drug articles, and it can happen the reverse way as well. See, the stability shelf life has been particular product or test article, but something happens during, like for example, it has happened to emergence of in many products, and it has come to the light midway during the study. So there is in the stability, and instead of extending the stability has to be reduced. So there are lots of permutations and combinations, and they need to be. The control samples should be retained for all studies except the short-term studies, like for example, SOPs. See, there are various documents which exist, but I have clubbed them right now. work instructions, the rules, the notices, the guidance, the quality manual, the policy manual, and other aspects of facility. And there are standard SOPs for particular function. For example, again, example of for and work instructions for use of mass spectroscope. There could be some particular arrangements to be done, instrument setups to be done for a particular study. This can be issued along with SOP for mass spectroscope as work instructions. This tell employees which action to take under variety of circumstances, whereas work instructions are very specific. Approved by test facility management. I already covered this aspect in details earlier, and so also revision should be separate test facility should have immediately available current SOPs. I mean, each test facility should or unit SOPs relevant to their activities being performed. The SOP should be based on published textbooks, analytical methods, or other aspects, and they should be mentioned in SOPs. I'm not going to go in details like what the content of SOP should be. That is but SOP deviations should be documented and should be acknowledged by the study director and PIs and sponsors and regulators. The SOPs should be available for, but not limited to, the following categories of test facility activities. See, these are subtopics: test and reference items, receipt, identification, labeling, handling, sampling, the apparatus use, maintenance, cleaning and calibration, and other things, material, reagent, solution, preparation, labeling. Then record keeping, reporting, storage, data collection, preparation of reports, indexing systems, handling of data, including use of computerized systems, room preparation and environmental room conditions for the test system. For example, how wet samples are going to pathology samples are going to be processed. All this to be documented in form. Receipt transfer, proper placement, characterization, and identification of test and reference systems. The test system should the test system preparation, observation, and examination during and at the conclusion of studies. Like for example, there is a quarantine involved in terms of, so these all should be mentioned clearly. Sample collection, then identification and hand. Group C and histopathology, for example. So all these aspects should be detailed in SOPs, appropriate SOPs. Separate SOPs should unit operation. I have a practice of I use MS Visio, which generates charts, flow chart. I sincerely believe that if we prepare one SOP for only one chart, for example, if particular activity one single flow chart, then we should probably break into sub parts where we can have say 2 or 3 or multiple, and then each part is described in details. So whenever we train people, they refer to both the and also the visual impact of the flow chart. So whenever the staff actually performs the and he accesses the flow chart, it is easier for him to correlate and remember things. At the same time, the unit operation approach also helps writing simple SOPs. The systems in a test plots, how they are going to be kept and handled in the test facility. The quality assurance procedures and processes should be in the form of SOPs. Operation of quality assurance people in planning, scheduling, performing Let's come to now performance of the study. The study plan. For each study, a written plan should be approved by dated signature of the study director, GLP compliance manual, personnel, key personnel, sponsor, if necessary, facility management, and it may get very committee wherever is appropriate. Amendments also pre-planned process, which is again written in the SOP, and any deviations in the study plan should be discussed and approved by the study director. There should be either intimation or again re-approval by the regulatory authorities, depending on the quality of these changes. Study plan should be the sponsor's name, dates, and corresponding numbers. There should be identification about the nature and purpose, identification systems for test and reference items, the details about test facility, study director, PI, study personnel, and scientists which are involved in the study execution should be mentioned in the study plan. The experiment and completion dates, the test methods, how in reference to OECD or any there are any particular issues with their applicable, for example, any unforeseen event or system, why and how, when and where should be justified, such as species, strain, substrain, source of supply, number, body weight range. In a clinical protocol, it will be a little bit different, the information, but the characterization is must. The method of use of test art and reference articles, administration involved in why intravenous or why oral, all these data should be part of experimental design, the descriptive chronological processes, all the methods, types and frequency of analysis, measurements, observation, and examinations to be done, performed, whether when the samples are to be, how it is to be collected, any statistical analysis to be done, if there is any coding or blinding which to be done for statistical or any control studies, say single blind or double blind studies. And these studies need not be necessarily only clinical, but even GLP studies can be blinded. And if the required to be open, then there is a correction in the statistical error, the alpha, and the acceptability of results becomes much more stringent if blinding is to be revealed for some reason, to be captured in the protocol, and details about the records as well. I think I can self-explanatory. The study has to be, of course, conducted in accordance with the study plan. Any deviation should be recorded. Specimens and everything should be stored and archived if necessary. All the data generated promptly, accurately, and legibly by individual entering the data. So this is extremely important. These are part of the good document. Each and every guideline. So these entries should be signed and initialed and dated. So any change in the, here again the quality important, because if there are forms, the data recording forms which are being issued by quality assurance, they should not get duplicated. committed a mistake and he is scared about his mistake and wants to hide those mistakes. He'll generate another form, and again this happen, and how to control this activity is a quality assurance perspective. So as not to obscure the previous entry. Suppose there is an entry error made. Instead of writing 12, person has written, it should be done in an appropriate manner following what is called as a good documentation practice, and the data generated as a document input should be identified at the time of data input by individuals responsible for direct data entries. So for example, pharma or SAS analysis or computerized systems, the full retention of full audit trail has to be there. You see, in mass spectroscopy, audit trail which happens at the time of entire analysis, during pre-sample and calibration, everything gets recorded, and whatever even the analyst opens the sample loading area, even that gets recorded these days, and why he had to open the sample he has to even and all this will get computerized, captured in audit trail, and that should be possible to associate all changes of data with the persons having made those changes. For example, use of time and dated signatures. Results for be checked by the next level of superiority. Conduct of study should be based on acronym. The data should be attributable. This was very more appropriate done with the handwriting, but it still is applicable even to computer system, because we can still create illegible. So attributable, legible, contemporaneous, original, and accurate. See, these are the main Alcohol plus, which has become dominant, and the data has to be completed. So like what I say, I will say truth, like the oath we take in the court, and not half truth or not half, not hiding part. It should be complete. It should be consistent. I can't add data from two different time frames or experiments, or to do two different unrelated work areas in one area. So consistency related to the work has to be there. Enduring, the data, where the data managed cannot be saved, and it should be made available to all concerned people, the quality assurance people, the study director, the regulators, as per their confidentiality and accessibility option. And these are the implicit basic ALCOA, and form the good documentation practice, and they're inherent in all. So this part is not exactly the GLP, but it has become implemented. It has become over the time inherent in any of study results should be prepared for each study, whether it's study completed, rejected, or whatever. The moment work is started, and report should be prepared. Reports of principal investigator or scientists involved in the study, they have appropriate levels, either study director or principal investigators, or all of them, or even some ethics committees do get involved in part of the activities if they are roped in between because of safety concerns and immediate reporting, or there is safety committee reports. And this is of course not directly related to GLP, but I'm just all reports should be signed and dated by PI and scientist and study director. The final report should be signed and acceptance of responsibility. The extent of compliance. Here, there is no, I can't say I'm 20% compliance or 80%. I have done my best. Is now yours to review. Additions to the final report, they can happen even after the release of final report, and they should be in the form of amendments, predefined manner, and with specification of reasons for corrections or additions, and final report to comply with the submission requirements of national registration or regulatory authorities does not constitute correction, addition, or amendment to the final report. Content, title page, all information which is very similar to the protocol. Same things again, whatever is, but it's a summary of results, then detailed information of all the data which is there as per protocol, as results including calculations and statistical significance and discussion of results, conclusions, and then archive where samples are stored, reference and test items are returned or stored, and or how they are archived, specimens are archived, and how they were destroyed. The raw data, where raw data is stored, because raw data is going to be much huge and larger than final. Going to be included in the final report, but how they are stored and how that can be accessed be mentioned. Okay, let's come to this aspect. Study plan, raw data, samples of test and reference, specimens, final report of studies, QA, as well as master schedules, records of training or people's detail, their CVs, their training, their experience, their job description, calibration of apparatus. All this should be archived appropriately. The validation documents of computerized systems or equipment, their earlier versions, their current versions, the impact analysis, if at all, environmental monitoring records, so temperature records, laboratories, or in individual cages, everything should be archived, and care should be taken so that these are preserved for the stipulated period of 15 or 20 years, as the case may be, or as per the agreement requirements. The final disposition of any study material should be documented and as per the approved SOP. And pre-planned entities should be involved. If sponsor is required to be involved, he should be involved. If he is not required to be involved, in destruction should be done in a proper documented manner. Only authorized people by management should have access to archives. So the moment of archival, people just cannot just walk in and actually access the study records. That cannot be possible, because once the study is completed, it becomes the management's responsibility and the study director's responsibility. It is their ownership. The study person, after studies completed, for accessing the data, because that is absolutely actually particularly no, because there's a big question, and then lots of permissions will be required if he really has to access that data. So the data, once it is locked, is, and very few people have access to this data later on. And if it is contracted out, and that particular test facility or archive goes out of business, how the data should be transferred to another facility should be pre-planned. I mean, so even at the study plan, it should be mentioned, be SOP in place. So that completes a lot of data. But let's see now compliance perspectives. So we all are in a meeting to discuss that why work is not being done. No, we should not be doing that. And because See, we are conducting an audit to check for compliance to the remarks in the audit conducted to check the compliance. And it cannot happen all the time. So this is a vicious cycle. But if you make it a routine and sin, this will never happen. There will not be a circular phenomena to understand why work is not being done. That will never be a compliant system and sincere and right from top to bottom. So to ensure compliance, what we need to do? We need to execute protocols using these quality systems. So quality systems, of course, do involve many things which will and everything. So QA, quality control and assurance. Now we'll see what is the difference between that. There is a monitoring activity. See, monitoring activity has a in GCP, and when it becomes sponsor, comes to sponsor, the monitoring and auditing are different. Then there is a review. There, all these activities are conducted to ensure compliance, and they are done by different people. See, building quality system management, test facility management, protocols again with the help of sponsor, regulatory authorities is done by the facility organization. QC and assurance test facility, monitoring by both test facility and the sponsor. Sometimes even the regulatory people, same test facility, sponsor, regulatory. Review is done by the generally by sponsor and is generally done by regulatory authorities. So, but everybody does what they want to ensure the compliance. Now let's see what is the difference between. Very few people illustrate this difference in my experience. See, quality control units responsible for inspecting and certifying a predefined quality expected in a product, instrument, or whatever process through quality control system. So there is a predefined quality expected specifics. So it means there is a specification document existing, and we are testing against that specification for compliance with that. Once that happens and all the specifications are complied, we say it's okay, acceptable. Whereas quality assurance done by audit or QA group, it assesses the performance, accuracy, reliability quality system through independent auditing activities. So quality control is part of quality assurance. Individual item, whereas quality assurance is a larger perspective which encompasses the entire multiple aspects, and it grows more across. Not, it is not related to one study, but it is related to the organization, multiple aspects, multiple people. Everything is controlled in quality assurance. For example, if in a particular study, see performance of different groups within their organization, they can decide to do one analysis. How many or how many kappa actions were required to be taken in each particular study. So there are 10 investigators in there, and they can say that each investigator has done 3 or 4 studies, and how many kappa activities were required to be done, how many errors were part of the quality assurance, and if a particular investigator has actually been able to control required in a study from 10 to 3 or 10 to 1, he has performed very well. Can't be 100% compliant all the time. There is given, there are going to be mistakes, there are going to be situations which need to be dealt. But quality assurance helps us in that. Monitoring is the act of overseeing the progress of a research study being conducted, recorded, or reported in accordance with protocol or SOP and GCP, GLP This definition I taken from GCP, but I always add it in GLP presentation as well because it's very important. Done by quality assurance people while they are doing the routine work, and it can be termed as a quality control assurance. Audit is again it's a systematic and independent examination of a research trial, and the record are being accurately reported per protocol or SOPs or good practice requirements. So inspection again is the same thing, but done by regulatory authorities. Ultimate aim. What is the ultimate aim of all this? The inspection by or audit or monitoring activity done by internal or external or regulatory authorities. Report to comments and action taken reports. We have to avail the clear certificate. And well, this means compliance. We are compliant. Compliance certificate is a that audit has taken place, evaluation has taken place, and the compliance, there were issues which were compliance to the SOPs, the compliance to the protocol, all details. It's just how inspector can find out policies. It's a very simple example I'm giving. So this is there is which is one digit, so minus 67.8. And in the log entries are 67.80. The person was returned 0 for no reason. So subsequently the recording was there, so there must be some QA they saw this anomaly and they changed it to again single digit. So there is sponsors monitors point of view, sponsors and auditors point of view, the inspector's view, how they're going to see this and what level they're going to dig this, why this happened, training, who did authorize this change. First of all, why the change was, why there was erroneous entry, training, there was a person who was qualified who did. All these questions will come up by just simple, this is 1 0 being added and it has generated. So participants is in compliance, like we check, it's a management of all organization. CROs, commercial, academic or other, all the employees, contractors and subcontractors, and regulators. Even regulators have whatever practice, GXP practice they are inspecting. And let's see one cartoon. Who met in a first class of a airline. Okay, now we can only dream for this, but as of presentation and those were the traveling days, a person looks very dismayed and he said, fellow passenger, it is very company like mine, very muddy office culture. There are no management information system. There's no team spirit, only leg pulling and whatnot. The other fellow says, "Oh, don't lose heart. You can try to change the job." But the person says, "Well, I can't. After I own this company." So it's a and it starts with the top management. Very important, and it is not an individual responsibility. Person has got a visitor in terms of sponsor and he's showing him around, and there are many more senior people, other things, and only one person who's working. See, these are all managers and this is our staff, and the staff is all responsible for compliance. Every person is responsible. See, again, repeating, compliance is an organizational responsibility and be in a serious business of data which is being submitted to regulatory authorities. Compliance is not an individual, organizational, and everybody is involved. Integrity is a culture. It cannot be as a part of and it's a continuous process. So document properly what you do. Do not document what you do not do. And line which I learned during my factory training in my pharmacy education. That it was written by a person in QC. He was very fond of this statement, and he had written it all over in the labs for the first time at right time in right manner. That is what is crux of any compliance. If there are critical deficiencies, I'll go through this very quickly. But what is okay? We can see the critical and major deficiency and other deficiency of error findings by regulatory authorities, and so also by even all organizations their findings in a similar manner. So, of course, deficiencies are going to be in higher numbers than critical. This is a 2017 and 18 data from Europe. And typical findings include the QA inspections not done, CAPA is the training records, or there's a conflict of interest. The equipment calibration is not done, performance is not appropriate, maintenance is not done. Study conduct related things. See, I mean, I can give some examples. Like we were auditing for a sponsor studies submitted by one sponsor through one CRO, and when we with team of we found there that during the trainings were not conducted at all. They were just written down, and somehow for us to find out because the records were written in a very illegible of handwritten and not recorded in an easy manner so that we can analyze. But somehow we were able to analyze that data, and it happened not on one occasion, multiple occasions. Many people in the facility were present in training, 3 different trainings. So no, that's how it is a serious thing. Training is a very serious thing. It has to actually and not just written down. Training is very neglected area in most of the organization. Even is not given due importance. But there are good CROs. I worked in such where at least 30 to 30 invested in training and retraining for the people, and it's a huge money is required for training, so it's not an easy or a cheap option. Compliance, the equipment calibration and performance and maintenance and archives related findings and study conducted and deficiencies, and I don't know whether it's very clear. Those archives related, there were around 50. Then computer facilities related, there are around 50 observations. Then organizational and personnel related, there are around 75. And then QA related, there are around 50. The most were study conduct related. So there are about 200 observations or deficiencies found in 469 inspections. So again, this is aspect, because whatever we are doing so that all the systems and SOPs and QA and study director and doing so that we can conduct the study properly. But despite all these systems, even in 2017-18, more conduct. So we have long miles to go. I'll not read this. This is actually over here. This is international status of GLP, and they're very similar in content and they differ jurisdiction differs under different government agencies. Like Canada in its health protection, and in GLP direction, and then FDA. There are two different departments which are involved. One another is which one? CFR. Okay, one is Title 21, and another one is by under EPA. There are important regulations which I am mentioned over here. This is of course is by WHO. There are directives of by European Commission, CFR, OECD documents, then revised guidelines for compliance monitoring procedures and applications of things. I'll not mention. Okay, here it comes to international GLPs. Okay, this is France. There is OECD, there is UK, there is US, multiple agencies, as I was mentioning, and Japan again. More countries. I will not go through this. There is India and GLP. Let's talk half a minute for this. DS has brought national coordination of testing and calibration facilities NCTCF in 80s, and it was in and in 1983 GLP concept was initiated. Initially, we were not the full member; we were only observers, I believe. Then we have worked similar OECD principles. There is National GLP Compliance Monitoring Authority also. In first GLP came in 1992, second in 1996. Both were toxicology labs, and interest for GLP in India. There are today more than 50 laboratories comply with GLP. We have become a full member of mutually acceptable data in OECD working group on GLP. So GLP Schedule L1, this is a part of the drugs and cosmetics rules, which is official and mandatory for all pharmaceutical laboratories, manufacturers, loan licensing units, and all with the aim of assuring true value of sample under the test. The Schedule 11 guidelines look brief in many guidelines, but still they are important. Now let's come to the concluding remarks. Oh, good, we are on time. So GLP pivot has a pivotal role in safety, validity, and reliability assurance, which directly contributes to the public health. So whatever we do is ultimately going to affect public. So GLP plays a major role in research, right from the development of a molecule to the design of proper dosage form, then in preclinical. GLP plays, whether compliance is required officially or not is a different aspect, but principles of GLP everywhere, even in research. So focus of GLP is to provide the principles or guidelines rather than detailed laboratory requirements. So pertinent to the modern laboratory changes. See, but see why GLP is really still active, because it is not very specific, and it is just a tool which we need, which we use. So spanner is like a spanner. Any nut in any equipment. So the equipment may become very, very complex. The shape of nuts may change. The spanner, the functionality of spanner remains always. So we may have an Allen key instead of those 10, 12, 18 numbers of spanner. They will be smaller nuts, but then functionality of spanner remains the same. The few future challenges in this line are perspectives are, of course, the electronic record keeping. So we are now going from paper to paperless. So how this transaction is going to be happening? Not only that, electronic data capture, EDC, everything is going to be EDC. There is no handwritten. We have already facing some this in good clinical practices in terms of electronic informed consent. And see, there is going to be issues related to connectivity. We had incidents in the morning for us as well. So nanotechnology products, which are because products are very simple, syrups, tablets, and other things, or powders or injections. But now there are different products. There are going to be devices. There are plant incorporated protectants, bioscience. Have some impact on GLP and how it is going to be implemented. And studies which needs to be taken into consideration. See, GLP confirms the good quality of the data on which regulatory authority can rely genuine results of case study. In lab can be done by an individual person for his own reasons, or at a study director level, or at the institute level. See, these frauds are it's a unavoidable human nature, and we need to curb that tendency at all levels. That's the basic purpose of GLP. The main purpose of GLP is to verify if is valid or not. And so by implementing GLP, even if a fraud is committed, then it gets surfaced of the GLP without the GLP system. It is very easier to catch frauds and misconducts. It also ensures that the quality data is submitted to the authorities and to public at a later stage. Country now for mutual acceptance of data in OECD working group. So GLP certificate from National GLP, NGC, EMA, and DST can be availed. So image of a company as a quality producer in the global market, it brings business. So GLP, everything we do is for business. It's all money, money, money. But at the same time, we have owners or responsibility for the people and our own integrity. It always playing a role in this compliance business. So these are the major references which are WHO GLP training and drug and cosmetic rules. But again, I'm putting these three circles. We are not looking at individual guidelines or compliance rules, but together we are actually going for a global picture. So this is actually the global by this circle around this many circles, and we have to create a very nice picture, not like a haphazard picture in the first. Don't have to we have created a good design which is very nice feeling to the eyes. So that's what the compliance is. So we can have a small question and answer if possible, or we can do it at later stage. And
thank you, Prashant Bode.
We'll reassemble at
Thank you, Prashant Bode. We'll break for lunch.
Yeah.
We'll have the question answer session at the end. And now it's 1:15.
Okay.
We'll meet at 2:15. All the colleagues, please let us meet at 2:15 without fail. And in fact, you can keep it logged in so that you need not get connected once again and have any problems. Prashant Bode, you please leave it in this condition. Leave it in this condition. We'll meet at 2:15.
Yes, folks, some of you may have to disconnect because you're using on phones and you're going somewhere else. But still, Please try and log in by 2 o'clock.
Yes, I will
request you
remain
to
calendar.
please log in by 2 o'clock. Yeah.
Yeah. Okay.